Dear students, Assalamu alaikum. We continue with the study of the white blood cells. Today we will start with a condition which is known as leukopenia. This is a clinical condition in which the bone marrow produces white blood cells lower than normal. Now, this leaves the body unprotected against many bacteria and other agents. Now, what happens? Normally in our body, all the mucous membranes of the body are constantly exposed to large number of bacteria. Effects of leukopenia. So when the body is left on the mercy of these bacteria which are present at different sites, what happens is that the ulcers may appear in the mouth and colon, or the person might develop some form of severe respiratory infection. Now, the bacteria from the ulcers rapidly invade the surrounding tissues and the blood, and without treatment, death may occur in less than a week. Causes of leukopenia include irradiation of the body by X-rays or gamma rays, exposure to drugs like chloramphenicol, thiuracil, exposure to chemicals that contain benzene or anthracene nuclei. Now, these can also cause aplasia of the bone marrow. Treatment of leukopenia. After moderate injury to the bone marrow, some stem cells are capable of regenerating the bone marrow, provided sufficient time is available. A patient who is properly treated with transfusions and appropriate antibiotics and other drugs to combat the infection usually develops new bone marrow within weeks to months for blood cell concentrations to return to normal. Another clinical condition related to the white blood cells are the leukemias. Leukemia is greatly increased numbers of abnormal white blood cells in the circulating blood. It can be caused by mutation of a myelogenous or lymphogenous cell. Types of leukemias. Leukemias are divided into two general types: lymphocytic leukemias and myelogenous leukemias. The lymphocytic leukemias are caused by increased production of lymphoid cells, usually beginning in a lymph node or any other lymphocytic tissue and then spreading to the other areas of the body. Myelogenous leukemia, on the other hand, begins by the production of young myelogenous cells in the bone marrow spreading throughout the body. So these white blood cells are produced in many extramedullary tissues like the lymph node, spleen, and the liver. So these leukemias may produce certain effects on the body, which include invasion of other body tissues, development of infection, severe anemia because the red blood cells, they are also decreased, bleeding tendency caused by lack of platelets. These effects result mainly from displacement of the normal bone marrow and lymphoid cells by the non-functional leukemic cells. And finally, an important effect of leukemia on the body is excessive use of metabolic substrates by the growing cancerous cells. The leukemic tissues reproduce new cells so rapidly that tremendous demands are made on the body. And these cells use the body reserves for foodstuffs, amino acids, and vitamins. Consequently, the energy of the patient is greatly depleted, and excessive utilization of amino acids by the leukemic cells cause rapid deterioration of the normal protein tissues of the body. So this results in the metabolic starvation and finally can cause death of the patient. We now move on to the next topic, which is the immunity. The human body has the ability to resist almost all types of organisms or toxins that tend to damage the tissues and organs. Now, this capability is called immunity. There are two types of immunity: innate immunity and acquired immunity. The acquired immunity is further classified into two subtypes called active immunity and passive immunity. You can see in this flow chart how immunity can be divided into its types. First, into innate immunity and acquired. Acquired is further divided into active immunity and passive, and active immunity is further divided into humoral immunity and cell-mediated immunity. Now let's first study the innate immunity. The immunity resulting from general processes is called innate immunity. And these general processes include, number one, the phagocytosis by the neutrophils and the macrophages, as we have already studied in the previous lectures. Secondly, the destruction of swallowed organisms. This is by the acid secretions of the stomach and the digestive enzymes. Number three, resistance of the skin to invasion by the organisms because of the protective layer. Then, presence in the blood of certain chemicals and cells that attach to the foreign organisms or toxins and then destroy them. These substances include lysozyme, which is a polysaccharide that attacks the bacteria and causes them to dissolute. Basic polypeptides, which inactivate certain types of bacteria, the complement system, and the natural killer lymphocytes, which can recognize and destroy the foreign cells, tumor cells, and even some infected cells. The innate immunity makes the body resistant to certain diseases, as the viral infections of animals, cattle, which may kill the animals, but the human body is protected. Now, similarly, many animals are resistant or immune to the human diseases because of their innate immunity, like poliomyelitis, mumps, human cholera, measles. Now these are very damaging and lethal to the human beings, but the animals are immune to them because of the processes of the innate immunity present in their bodies. Acquired immunity. The ability to develop powerful specific immunity against individual invading agents is known as the acquired immunity. Now remember, the key features of immune response in the acquired immunity are: number one, specificity against the specific organisms, memory, and clonality. Clonality is the formation of different clones, as we shall study in the next lectures. As mentioned earlier, there are two types of acquired immunity: humoral immunity and the cell-mediated immunity. Humoral immunity is also known as the B-cell immunity, and the cell-mediated immunity is known as the T-cell immunity. Now, before moving on to study these types individually, let's first study the role of the antigens in the immunity. Now, both types of acquired immunity are initiated by the antigens. Now, what are antigens? Technically, now each type of organism almost always contains one or more specific chemical compounds in its structure that is different from all other compounds. Generally, these are proteins or large polysaccharides, and it is these that initiate the acquired immunity. And these substances are called antigens. And for a substance to be antigenic, it usually must have a high molecular weight of 8,000 or above. In addition, the process of antigenicity also depends on the regularly recurring molecular groups on the surface of these large molecules, and these are known as the epitopes. So epitopes are present on the surface of a large molecule in a foreign organism, and because of the presence of these epitopes, that molecule is designated as an antigen. Now, as in both types of acquired immunity depend upon the presence of the lymphocytes, let's have a little review. of the lymphocytes. So acquired immunity is the function of the body's lymphocyte, and acquired immunity cannot develop in people who have a lack of lymphocytes. The lymphocytes are located extensively in the lymph nodes, and they are also found in special lymphoid tissues, which include the spleen, the submucosal areas of the gastrointestinal tract, thymus, and the bone marrow. The lymphocytes are divided into two main types, the T lymphocytes and the B lymphocytes, and we call the immunity which is produced by T lymphocytes as the cell-mediated immunity, and the immunity which is the function of the B lymphocytes is known as the humoral immunity. Lymphopoiesis. Both types of lymphocytes are derived from the pluripotent hematopoietic stem cells that form common lymphoid progenitor cells, as we have already studied. The first cell in the stages of lymphopoiesis is the lymphoblast. It has a size around 15 to 20 micrometers, contains a large nucleus. The cytoplasm is bluish and appears as a thin rim around the large nucleus. The next cell is the pro-lymphocyte, in which the nucleus becomes smaller and the cytoplasm is less basophilic. The mature lymphocyte has a size around 10 micrometers or less. The nucleus is deep blue and very easily recognizable, and the cytoplasm may be in the form of a thin rim or maybe abundant. Now these, you can see in these pictures, a lymphocyte present amongst the red blood cells. Preprocessing of the lymphocytes. Now, before the lymphocytes they move on to the lymphoid tissue and perform their functions, they have to be preprocessed. And the two main steps in the preprocessing, one is the non-antigenic development and the other one is the antigenic development. So we'll first study the non-antigenic development, and as I said, that all of the lymphocytes, they eventually end up in the lymphoid tissue, but before doing so they are to be preprocessed by these processes. So we'll continue with these processes involved in the pre-processing of the lymphocytes in the next lecture.